Hi, I'm Matt Smith, and I'm going to be presenting today on machine learning classification methods for single molecule peptide sequencing data. In particular, we're looking at fluorosequencing sequencing data. This has been under development in my advisor's lab for several years, this technique. It's useful for identification and quantification of proteins, and it's particularly valuable for post-translational modification studies. In fluorosequencing, you start with a biological sample of proteins, and you may not know what all proteins are in that sample, and you take these proteins and you're going to denature them or unfold them into a string of amino acids, where each of these letters here is one amino acid. We'll then apply a protease to this, which will cut up this string of amino acids into smaller chunks, which we call peptides. We then add fluorophores to this, which are designed to label specific amino acids. So here, for example, the K, which stands for lysine, is labeled by blue fluorophores. And we'll then take these peptides and anchor them to a flow cell by their C terminus. When we do this, we can look at the flow cell under a microscope and get a bunch of bright white spots. These bright spots are each one peptide with fluorophores on it, and we can perform Edman degradation on the flow cell, which will remove one amino acid from the N terminus, and doing this successively, we can sequence a lot of peptides in parallel. Once we've done this, we need to analyze the data computationally. This has a couple of steps. The first is to follow each of these bright spots across different colors of fluorophore and across time to find a fluorescent read sequence of these intensity values representing how much light there is in the image. And then from that, we want to find what is the peptide that produced this data. And if we know the peptide, that hopefully tells us something about the protein that was in the original sample. Today we're going to be focused primarily on this second step, going from a read to finding the correct peptide. So this may seem easy at first. It's not. This is a histogram of intensity values, and here we've done a fit of a mixture distribution for different numbers of fluorophores, and you can see that there's a lot of overlap, which means we don't really know necessarily exactly how many fluorophores are at any point in the image at any point in time, and so this is a little more chaotic than we'd like. We also might have Edman degradations fail to occur when they're supposed to. This happens about 6% of the time. We also lose fluorophores while we're doing sequencing. We might have fluorophores missing before we even start sequencing. And when we're trying to do sequencing, the peptide might pop off the flow cell. And so all of these forms of error serve to make this a lot more difficult than it at first appears. So one thing we might try is to use some kind of standard machine learning classifier. One example is k-nearest neighbors. Here, imagine we're trying to classify this purple question mark. What we would do in k-nearest neighbors is essentially ask, what are the nearby data points classified as? And in this case, the nearby data points are all blue bunnies, and so we would probably assume that this purple question mark is a blue bunny. There's ways to do this with more mathematical rigor, but this is the basic idea. For our problem, we're dealing with a much higher dimensional space. For example, here we've got 9 time steps and 3 colors of fluorophore. That makes this a 27-dimensional vector of data for this read. But that doesn't really the algorithm is still something we can do. We just look at similar reads and classify based on the reads that are the most similar. When we do this, we can get precision and recall curves. If you haven't seen a precision and recall curve before, just know that more area under the curve is better, so that's our goal. And then we can also measure the runtime. Here this is about a minute. So this is read recall, and then we've done some really basic protein inference to give a protein recall down here on the data set that we're using. This is simulated data. The whole human proteome has been trypsinized. We've selected 100 proteins randomly and labeled 3 colors of fluorophore and simulated 10,000 sequencing reads. So this is okay, but we'd like to do better. So what else can we try? Well, we'd like to just know, hey, what's the probability of this peptide from this read? But we can't get that. And maybe what we can get instead is to find what's the probability of this read given one of these peptides. And if we can do that, we can use Bayes' rule to flip this around and get the probability value that we want that will be more useful for classification. So how do we do that? What we can use is a hidden Markov model. And in our hidden Markov model, we need states to represent our sequencing conditions. And so we need to deal with different numbers of amino acids being removed during sequencing and different combinations of fluorophores being present or not, depending on which ones have been destroyed. And so if we do this, we also need transition probabilities between these states. And so this is what that looks like. It's a little bit messy, but let's not worry about that yet. Not all transitions are possible, but there's still a lot of possible transitions. But this is basically the model. And when we have a hidden Markov model, we can run something called the forward algorithm, which when we do that and we sum up all the states at the end, we find the probability of a particular read given a particular peptide. In this case, the peptide right here. So this approach has some challenges, and we're going to go through these one at a time. The first challenge is that larger peptides have way too many states. So for larger peptides, we need to have states for every fluorophore being there or not in any combination. And so if you have 10 fluorophores, you have 1,000 states for that. If you have 20 fluorophores, you have 1,000,000 states for that. And so it grows completely out of control. And so what we want to do is merge together states that are very similar. And we're going to merge states that have the same number of amino acids removed and the same counts of fluorophores of every color, even if they're not labeling the same amino acid. So here are these two conditions. A different lysine is labeled on this peptide. And we can do this with no loss in accuracy of the model. To see that, first, let's take a look at this without Edman degradation. And so if we do that, that's relatively straightforward. The position of these fluorophores doesn't matter. We can't actually see any difference under the microscope, and so it all comes out the same. If we do one more step of Edman degradation, this N-terminal amino acid, this arginine here, doesn't have a fluorophore on it in any scenario, and so we still have no loss in this model. If we do one more step, then we have to start thinking about where our fluorophores are. But since we expect that these fluorophores should behave equally, then by symmetry, we should expect that we have an equal probability of being in this state or this state. And so if we have a successful Edman degradation event, we would then expect that there's a 50% chance that you lose a fluorophore and a 50% chance that you don't. And this can be generalized for higher numbers of fluorophores and things like that. But that's kind of the basic idea. So we can expand this on this to fill out our model for this particular peptide. And so that's kind of how we deal with the massive state space of these peptides. Another issue we need to deal with is that the transition matrix is very dense. And this is a problem because, and so each of these black arrows here is a non-zero entry in the transition matrix, and we're doing the forward algorithm, which means we need to multiply by this transition matrix over and over and over again. And we want something more sparse. One way to do that is to factor this matrix into a product of matrices that have a greater sparsity effect. And what we can do is we break it down by different types of effects. And so we look separately at the blue fluorophores, the green fluorophores, Edmond degradation, and peptide detachment. By viewing these all separately, we greatly simplify the problem because we no longer have to think about combinations of events. We don't have to have a transition probability specifically for losing two blue dyes while not losing a green dye and succeeding in an Edmond degradation and not having the peptide detach. We just handle all of those independently and it comes out of the mathematics and we get the same effect as the original transition matrix. So that's kind of how we deal with the density. And this gets us to a point where we can ask how we're doing because we've gotten the problem to be tractable so that we can actually plot our situation. And so this is how we did with k-nearest neighbors, and this is how the Markov model does. As you can see, we have a lot more area under the curve, which is good, but the runtime is horrific. We're looking at about 3 hours here or something like that. So what else can we do? We can try to deal with this situation where we're wasting a lot of time calculating highly unlikely scenarios. And so the thing to realize is that while our intensity values have overlap for different counts of fluorophores, there's still values that are completely unreasonable for a particular count. If you're over here, a 3 count of fluorophores is not likely. And so then we're wasting a lot of time churning through transitions that are completely improbable. And so if we can cut this down to say, hey, we know we're at one of these states at this time step, and we're at one of these states at the next time step, then we only have to worry about the corresponding transitions. For larger peptides, this still grows on us in ways we don't want to deal with. And so we'd like to be able to combine this with that factoring that we dealt with before. And we can do that. And this is ultimately how we deal with this issue with all of these incredibly improbable states. And so let's see how much this helped. This is what we had before, and this is what we have now with this primed model. The precision and recall are virtually identical, and the runtime is about 5 times better. So one more issue is that we're running the hidden Markov model for every single possible peptide. There's over 1 million of them on the conditions we're showing in this presentation here. And part of the reason k-nearest neighbors does so well is that it scales logarithmically to massive data sets. But its results are far worse than HMM's, obviously, and so we kind of want to get the best of both. Can we have it all? So the trick here is to use both models. So we take our read and we first classify with k-nearest neighbors, and then we take maybe 1,000 of the best peptide candidates, and we're going to only process those with our hidden Markov model. And then when we do that, the hidden Markov model can deal with the weakness of the k-nearest neighbors, hopefully. And so that's how we deal with this. Let's see if it worked. So this is what we had before. And this is what this hybrid approach using both of these together achieves. The precision and recall is wiggling around on top of the precision recall from before. In either case, the reads and the proteins, and the runtime is comparable with nearest neighbors. I think it takes twice as long, but it's basically in the same ballpark. And so that pretty much concludes my talk. I'd like to acknowledge a number of people that have been helpful to me. Angela Bardot, Brendan Floyd, Jagannath Swaminathan. Eric Anslin is a PI in another lab that we work with. He's not pictured. This is Edward Marcotte, my PI, and I've also worked closely with Zach Simpson, who is not pictured. These are my references. Thank you for your time.